Okay, this lesson is about endo and exocytosis. We're going to talk about both in turn and some of the applications that are really kind of critical for cells. So, endocytosis first. The definition is it's a process where part of the membrane will fold in to form a vesicle inside containing a material that was once outside and move it into the cell. So some basics here. So you're going to see that there's a molecule, a larger molecule that kind of comes to the surface. You can see this like a little budding in, this bowing in. You form this little vesicle in here, and then it forms a what's called an endosome, and then eventually those things are broken down by these digestive enzymes, and this is what forms a lysosome. So endocytosis does require ATP, so we would classify it as active transport. Sometimes you can't just have it on the surface, but you need a receptor to pull it in. So we would call this receptor-mediated endocytosis. So you can see this molecule here, it's got to be stuck to it. And then that would pull it in. A lot of viruses will enter a cell through some sort of receptor-mediated endocytosis. A lot of white blood cells also use receptor-mediated endocytosis to attach the outside of their receptors to whatever bacteria or virus that it's going to try to destroy through phagocytosis. And speaking of phagocytosis, this is where a large molecule is going to be engulfed. Think white blood cells, they would engulf another foreign object. And then you have sometimes called penocytosis. This is where more water-soluble molecules are taken in. Sometimes phagocytosis is called cell eating, and penocytosis is called cell drinking. But not really a true statement, but it gives you an idea of like the size of the molecules being taken in. Okay, so what is actually carried in by endocytosis? You can have large molecules that can't pass through the membrane protein. You can have a larger material like a dead organelle from another cell to break down and kind of consume it. You can have large amounts of water taken in with the soluble particles that the cell needs. Exocytosis now. This is the opposite direction. Now we're pushing things out. So exo for exit, endo for enter. The process where a vesicle fuses with a membrane and releases whatever in the vesicle to the outside of the cell. And sometimes this is how a cell kind of replaces its membrane or adds to its membrane through this process. So so that you can see right here, the vesicle is getting closer and closer, and then eventually it attaches to and becomes a part of that whole cell membrane. So the vesicle membrane becomes part of the cell membrane, and whatever was inside is released out of the cell. This does require ATP, so we would call it an active transport. This is an important part. So this is how cells are going to actually build their membranes. You can see after, if you look at before endocytosis over here, let me go back here for a second, I apologize. You can see before endocytosis, you have this vesicle with these membrane proteins are on there, and you have this like little, like looks like some sort of glycolipid that's attached here, part saccharide and part lipid. And then when this membrane here would fuse just like this, so now what's on the inside goes to the outside. And then what does exocytosis carry out? A lot of like waste molecules, large amounts of it, larger particles like that. It also uses cell signaling molecules to kind of talk to other cells that have receptors that bind to it. Okay, that's it. Thank you.